All right, so before we get started on today's video, I do want to remind you that as you're watching the video, you are able to pause and kind of rewind back to wherever section you need to re-listen to or re-watch in order to get answers to help you answer the questions. So starting off, I would like you to ask yourself and try to answer this. There's no wrong answers, I just want you to try. Number one, how do babies grow in size to become adults? So pause the video and go down to number one and try to answer it there. Now, number two, what happens to the cells in your hand as you heal from a paper cut or any cut, for example? So pause the video and go down and try to answer number two. Okay, so we are going to go through the answers, and if yours are right, and then leave them. If they're wrong, then you can go ahead and correct them. So number one is, how do babies grow in size to become adults? Well, they're going to grow by multiplying their cells. Their cells don't actually get that much bigger because once you get to a certain size, the cells have to multiply in order to be able to maintain exchanging waste, exchanging gas, and exchanging nutrients. So the reason why we grow from babies all the way to adult size is because our cells are dividing. Now, number two, what happens to the cells in your hand as you heal from a paper cut? Well, we know that the cells might have either died or gotten injured, so the cells need to be replaced, and they're going to be replaced by the surrounding cells dividing and providing more cells that actually produce the scar tissue and the healing tissue that actually is going to form over where the cut is and then turn it back to normal. So our learning objective for today is going to be that students will be able to list the steps of the cell cycle and explain what is occurring in each step. So the cell cycle is about when living things are growing by increasing the number of cells and the size of cells. So like I said before, your cells can only get so big. If they get too big, then they're not able to exchange things with the outside environment as well, because as you can see, the nucleus gets really, really far away because the nucleus isn't going to grow in size, but the cell volume does. And so the larger a cell gets, the more demand it places on its DNA. And it's going to have a difficult time maintaining homeostasis, maintaining certain internal temperatures, maintaining enough oxygen inside, because it's hard for it to move stuff in and out of the cell. So the cell is going to have to divide instead. So before a cell gets too big, it divides. And then one cell will always become two daughter cells. So one cell in the normal cell cycle will split into two identical daughter cells. So why does the cell cycle happen? There are three major purposes of doing the cell cycle or going through the process of splitting the cells into two daughter cells. And the first one would be growing or growing larger, right? Like I said before, you're not growing because your cells are necessarily getting bigger. They are, just only to a certain limit, right? The reason you're growing bigger is because you're getting more and more cells. The second one is repair, like question number 2 from earlier. You're going to repair things that break or tear or rip by dividing cells that are healthy to fill up the empty space left by a wound. And the last one is going to be for reproduction. We're not actually going to talk about reproduction, producing sperm and egg in this current topic, but in one of the next units, we will talk about it. So again, the three purposes of cell division are growth, repair, and reproduction. Now, the cell cycle has two major parts. Interphase is when the cell is growing and developing, and mitosis when the cell is actually dividing. And as you can see in the diagram, most of the time, a cell is going to be found in interphase. Very, very, very few times when you look at any cells will you catch it in mitosis, because it spends about 90% of its life in interphase, and then about 10% of its life is going through mitosis. It's a very quick, very abrupt, very large process that happens, but the cell isn't functional during mitosis, so it's not going to waste time staying in mitosis for a long time. When it can get out, make the cells, and then those cells can go on and do their job while they're in interphase. So as you can see, we're going to start over here in this corner. The first part of interphase is actually divided into three parts, right? The first part is going to be the G1 phase, or the first growth phase, right? G for growth, 1 for first, right? The first growth phase is the G1 phase. And this is where cells are going to grow, they're going to make proteins or synthesize proteins, and they're going to produce RNA. The next phase, we're going around the circle clockwise. Here is the S phase, or the synthesis phase. And it's called the synthesis phase only because it's literally when DNA is synthesized, or when DNA replication occurs. Okay, synthesis, replication, duplication, they're all used interchangeably for the same meaning of more DNA is being made or DNA is being copied. Okay, so S phase stands for the synthesis phase. The next phase that you're going to reach And is the last phase in interphase is going to be the G2 phase, or the second growth phase, right? G1, first growth, G2, second growth, right? The cell is going to finish growing, and it's going to prepare for mitosis by making sure everything inside of it is doubled. The DNA should have been doubled earlier, so all the other organelles need to be doubled as well. And that's what the G2 phase is going to finish up. So why do cells perform DNA synthesis or replication? Well, they need to make an identical copy for DNA, because the new cell that's going to be made when this current cell divides into two is going to also need DNA inside of it, right? Because all cells require genetic information, which is made up of DNA. The new cell is going to need this DNA because it needs instructions for its purpose during growth, repair, or reproduction. If you don't tell it what to do, there's no point in making it to begin with. So when does it occur? We just talked about that. DNA replication occurs in the S phase of the cell cycle. S stands for synthesis, like I just said. So DNA replication is the exact same thing as DNA synthesis, and I will be using it interchangeably, so you got to get used to it. And the S phase is between G1 and G2, so never get that confused. Sometimes you're going to be like, well, why doesn't it make sense just to do G1 and then G2? Because it's 1 and 2, right? No, it just happens that the growth phases are on either side of the synthesis phase, because DNA synthesis is so important that they made its own section of the phase that is solely dedicated to it before the cell even finishes growing. So during DNA replication, DNA makes a copy of itself to prepare for cell division. An enzyme called helicase breaks apart the hydrogen bonds that connect the two strands of DNA like a zipper. So it's going to unzip the DNA. The easy way to remember that is because a helicase, it sounds like a helicopter, right? And another name for a helicopter is a chopper. It's literally going to chop down the middle of DNA and separate it like a zipper. These strands of DNA are called template strands because they are the template for making the other side of the DNA. So remember, now we're going to follow the rules of G always goes with C, A always goes with T, and new strands are going to be built based on these two template open strands. DNA polymerase is going to be doing that job. So as the old DNA unzips, an enzyme called DNA polymerase brings in new free-floating nucleotides that were in the nucleus. DNA polymerase matches base pairs using the unzipped DNA as a template. So it's going to use the unzipped DNA as a template and build this new strand on the inside, and it's going to go all the way down so that all of the DNA is copied. DNA polymerase also proofreads the new DNA molecules for errors. So just in case there's mistakes, it's going to go back and check itself once just to catch anything that could have been wrong and replace and fix all those mistakes. So it's like when you proofread an essay before you turn it in. An enzyme called DNA ligase then joins the nucleotides that DNA polymerase brought in. DNA ligase makes the nucleotides into a new strand of DNA. So it's basically going to glue all of them together into one big strand of DNA, so that now you have a completed strand of DNA instead of a bunch of little bases just sitting together. By the end, after replication is complete, there are two new double helices, right? They will recoil back into the helix shape. And we also need to note that DNA replication is called semi-conservative because half of the new DNA is actually made of the old DNA that was copied from the template strand, right? And then half is brand new, just built by polymerase and ligase. If you look, the new strands are actually still half blue when the old one was full blue. In a nutshell, helicase will unzip DNA. DNA polymerase will bring in and build new nucleotides. Ligase glues the new nucleotides together to form the new DNA strand, and then you end up with two identical DNA molecules that were semi-conservative or made out of half of the old DNA. And so we're going back to this picture now, and we talked about G1, we talked about S phase in a lot of detail, right? We talked about the synthesis right now, and then we also talked about the G2 phase of interphase. Well, there are checkpoints during these because you got to make sure that everything is going smoothly and that all of the requirements to move on to the next phase in the interphase and in the cell cycle in general are met before you try and go to do the next process. Because if you're missing things that you need for the next process, then something is going to go horribly wrong with the rest of the cell cycle, okay? So the G1 checkpoint checks to see if DNA is ready for replication. And the G1 checkpoint happens right here before you enter S phase, right? You're going to check first. Are we ready to prepare and go into S phase and to replicate DNA? So after you've done some growing and you've done some synthesizing of proteins, you're going to be like, are we ready to make new DNA? If not, we're going to pause here and recheck everything that we need to have done in G1. The G2 checkpoint checks to see if DNA has replicated properly. So as you can see, DNA replication is super important because you're going to check 2 times, before and after, if it happened right. So if you look at this here, you're going to see G2 checkpoint is going to be located right around here because you're checking right after you do the S phase if everything went right with the synthesis of the new DNA. And the last checkpoint, there are only 3 in the cell cycle, is the M checkpoint. And it's going to check if spindles and the alignment of chromosomes is going right at the beginning of mitosis. So right here in the entrance of prophase and metaphase within prophase of mitosis, you're going to be checking, hey, are things attaching properly? Is the setup going to go correctly so that we can prepare to do the rest of mitosis? We've already done our growing, our DNA replication, and some more growing and duplication of organelles. Now, will the split itself happen correctly? The M checkpoint is there to check if that preparation is going well. If you miss these checkpoints, then you can result in mutations, you can result in huge issues, you can result in cell death. So it's super important to have checkpoints to make sure that everything is going smoothly during the process of the cell cycle. Now, the cell cycle also has this little caveat right here of G0. And G0 is kind of like a resting from division place where cells sometimes stop at. It's kind of like a rest stop on a road trip. You're going through here and you're going through interphase. You're going to grow, you're going to duplicate your DNA, you're going to grow again and duplicate your organelles, and you're going to split. Sometimes cells don't need to split anymore, or there's no need for them to grow, or the body to grow, or for anything to be repaired, right? So some cells will enter G0. And G0 isn't dormancy, it's not resting, it's not really hibernation. It just means that the cell is not going to split. In G0, the cells are still doing their job. So the muscle cells are still contracting, helping with movement. The brain cells are still sending electrical signals for nerve endings to make sure that there's connections, right? So in G0, a cell is still doing its job. It's just not currently getting ready to split. So a lot of the time cells are in G0 when there's no need for them to be dividing their cell. And if there's a need all of a sudden because there's a cut or an injury, then the cell will get stimulated by either a hormone or some other outside factor to go back into the cell cycle and continue to replicate and divide.